right, so back to genome size as an aspect of phenotype. One way to change genome size is just to duplicate genes or get rid of copies of genes. And in general, a way that a variety of different organisms do that is with ribosomes. And so this is not a cell biology class or a biochemistry class. I'm just plopping this up here so that you're reminded that ribosomes are actually really, really complicated. But the important thing to remember is that they're the sort of molecular machinery of translation. And so the number of ribosomal subunits that you have operating at any one time within a cell will often be correlated with the sort of speed of translation, because if you have more ribosomes in a cell, then you can have more parallel translation projects happening at the same time. And so in general, if you think about different trade-offs between phosphorus requirements and growth rate, if you have really high selective pressure for high growth rate, there's a couple of different routes to that. You can reduce the exon count in your DNA. You can get rid of, you can reduce the intron count in your DNA. If you have selective pressure for high growth rate, or you can up your ribosome count and increase the rate of protein synthesis. So you can there's kind of different routes to the same thing. So you can have faster cell division if you have a smaller genome, but you can have a higher rate of protein synthesis if you have more ribosomes. And so depending on the nutrients that are available, you might, the trade-off might be more favorable one way or the other. So there's two different roads to higher growth rate that you can take. You can reduce your genome so you just have less information to copy, faster cell division potentially, barring the fact that you still have to acquire nutrients somehow. Or you can have higher rates of protein synthesis, which can sometimes in some nutrient conditions result in, actually end up resulting in, higher growth rate. So we can kind of visualize this trade-off a bunch of different ways, but one way of looking at it is just by ribosomal subunit copy number and the maximum growth rate. So regardless of nutrient availability, and in general, the more ribosomal units you have per cell, per genome really, the faster your maximum growth rate is. However, the maximum growth rate is inversely correlated with the carbon use efficiency, and that directly follows in many cases from the fact that ribosomes are energy intensive, and this changes the ecological stoichiometric balance of the organism. There's a couple of different reasons why genome streamlining in bacteria and other types of organisms happen. The two main reasons are because the bacteria are some sort of symbiont parasite or sort of intracellular commensal. I can't think of any examples of intracellular commensals, but in general, organisms that are symbiotic mutualists that live in other cells, or symbiotic parasites live inside other cells, they tend to have really low effective population size. This is an N sub E. And so in some cases, they just lose genes as a result of drift. And this can often result in the accidental, more or less, loss of genes, so that they have fewer than 1,000 in many cases. And they're really just reduced down to kind of the core metabolic requirements, the core metabolic repertoire. Pseudogenes can frequently be found in intracellular symbionts. Pseudogenes, I don't know if we've talked about that yet, but that's a gene that is no longer functional. And it's usually a step along the way towards gene loss. So in a lot of cases, if a gene is no longer under positive selection, it will undergo, sometimes people call this process pseudogenization, because it's making it into a pseudogene. And essentially what happens is there's deleterious mutations that cause the gene not to function anymore. And in many cases, this, you'll see stop codons pop up just like within the gene. You can also potentially see duplications in genes. So there's not always a straightforward path to genome reduction in all symbionts. In some cases, we'll kind of circle back to this. There are actually weird examples of chloroplasts that have like a bunch of genes, or mitochondria that have a bunch of genes. But in general, genomes tend to be streamlined in intracellular parasites. And then there's also, in certain situations, free living cells can have selection for genome streamlining if there's a lot of selection, like we talked about before, for high growth rate in the absence of high phosphorus. So if there's strong selection for high growth rate and phosphorus is really strongly limited, then the only way you can increase your growth rate is by trimming down that genome size. And so this is sometimes observed in planktonic bacteria in oligotrophic conditions where there's not a lot of nutrients in the water or in whatever medium they're growing in. Paralogization. I'm just gonna touch upon a couple of quick examples. These are in the book, which is, I think this is the first time that I've talked about the book so far. Paralogization, you can visualize the gene tree for paralogs as it's almost like an intragenomic speciation event, really. So this part of the gene tree is just two different genes within one super duper cute monkey genome. Whereas the rhesus monkey in humans, we only have one copy of these genes. But the genes that were duplicated in the langur, I don't know that monkey. The genes that were duplicated in the langur enable them to metabolize some bacterial byproducts, or that they enable them to take advantage of kind of more of like almost a cow style fermentation in their guts that other monkeys and apes can't do. So this is a situation where we can track the evolutionary similarity of a gene and its paralog. And in some cases, there's evidence of really strong high DN-DS ratio, which is consistent with the idea that there's positive selection happening. And in some cases, paralogs are actually more different from each other than the homologous gene is from the sort of homologous gene in other organisms. So in some cases, within one organism, it's almost like it's a speciation event, but just for a gene within a genome. I don't know if you guys remember these little puppies, but the mouse lemurs actually have some really super duper cool pheromone receptors that are associated with what's called the vomeronasal. Yeah, I always mess that up. But these are sort of intermembrane amino acid sequences, and certain parts of these amino acid sequences have been demonstrated to be more strongly divergent from each other than others. And these probably impact the different mouse lemurs' ability to react to pheromones. And there's a major proliferation of these essentially pheromone receptors in mouse lemurs. So these little bar graphs on different parts of this phylogenetic tree are trying to indicate how many different vomeronasal gene copies there are in the different genomes of these species. And so what they're showing in this phylogenetic reconstruction is that there's not as many copies of this, there's not as many paralogs in the hypothetical ancestral lemur or in any extant lemurs. But once you get over to the mouse lemurs, they just have a ton of these pheromonal receptors. And these are thought to play a role in speciation diversification dynamics of this group of organisms. And it probably plays a major role in mate recognition, species recognition. We're really still starting to understand what the sort of major big picture macroevolutionary patterns are of gene loss and gain in kind of like really big timescales. This is from a really recent study that came out just a couple of months ago, or just a month ago maybe, that looks at patterns of gene loss and then novel gene gain in different major lineages of animals. And the way to read this is that the sort of bottom two rows are showing the extent to which there's gene loss, and the more pink there is, the more loss there's been, the more blue, the less loss there's been. So what these authors have found is that annelids have lost more than 1,000 genes with respect to other animals. Whereas nematodes and mollusks have actually lost very few genes, it seems. And then the top two rows of boxes are looking at novel gene gain. And it seems as if nematodes have a lot of examples of novel genes in their genome. Tardigrades also have a lot of examples of novel genes. Whereas annelids have very, very few novel gene acquisitions. So this is sort of an emerging pattern that we're starting to see. People are still trying to make sense of. But I just thought it was really cool because it's a really recent paper. So I'm going to switch over to one of the last legs of our journey and start talking about inferring ancient whole genome duplication events in different types of organisms in the next video. So stay tuned for that. And yeah, keep washing those hands, y'all.